Hello everyone, this is Vaibhav. My project is synthetic block detection using sophisticates. Basically, synthetic blocks are nothing but conserved DNA regions across different species. And I am going to achieve this by using sophisticates through by building sophisticates using open and solid data. I am trying to find LCS and MUMs. And after finding them, I'm using some filtering and merging algorithms to find biologically important regions. And this is how I'm going to find secondary blocks. Other than that, I'm going to compare like how many insertions, deletions, and substitutions are required to convert this genome to other genome. And I'm going to visualize this outcomes as a dot plot and other visualization techniques. My contents in this presentation are introduction, project objectives, background, methods, current status, summary, and references. Evolution is a common phenomenon in our nature. We humans are also an evolution of an organism that existed in past. Genome aggregate mutations like insertions, deletions, and substitutions, which results in organisms that behave, act, and structure and are differently structured from each other. But there are some regions that are conserved in our DNA across species, and these conserved regions are called synteny blocks, which indicates common heritage. By studying on these synteny blocks, we can find how the evolution process happened. In this project, we are using a computational approach mainly based on suffix trees. Come to our project objectives. We are going to find synteny blocks using suffix trees. We are using concurrent algorithm to efficiently construct suffix trees. After that, we are going to extract longest common substrings LCS and maximum unique matches MUM. So after extracting LCS and MUMs, we are applying filtering and merging algorithms on overlapping or adjacent conserved regions in our genome. After doing that, we are going to calculate genomic changes or going to work on evolutions like how many insertions, deletions, and substitutions are required for a particular genome to convert into another genome. Uh, we are going to calculate this with the help of Lewinstein distance, and after that, we are also going to calculate uh, or tell how how much is the similarity between those two given genomes using Jagged index, and after that we are going to visualize using dot plot and synteny maps. Coming to the map, first we will go to why was the launch. What was the synteny maps of the conserved regions in the of different species. For example, consider human and chimpanzees. They both have common ancestors, but we live, act, and behave differently. This is because the mutation happened differently with that species and humans. It's a by studying synthetic we can find how they evolved differently and what are the factors that made them evolve differently. And by the study of synthetic blocks also help in comparative genomics. Coming to the computational techniques we are going to use in our project, we are going to build substress using components of algorithm. This algorithm helps to build substress and linear time, like time for which stays around week of time. And after that, we are going to extract LCS and appearance. Uh, by constructing substress, it makes it easy to find LCS and appearance. And after that, we are going to use jacket index to find similarity between two genomes and dimension distance to find insertions, deletions, and rearrangements that that happens in the given two genomes. ATP needs the chemicals. Some programmers created a tool called FOMA, which is based on sophisticated amines and is widely used for large unit comparison. And a researcher called McLean proposed a method called centenary mapping, which he used for detecting shared local intentions. And a researcher called Chen also proposed a method based on suffix tree for homologous secondary block protection. In this presentation, I came across suffix trees many times. On what is suffix tree? So a suffix tree is nothing but a compressed tree which represents all suffixes of a given string. It allows fast searching, pattern matching, and substring matching. I am using this method in this project to extract LCS and MMs, which in turn help me in getting to achieving my goal of centennial block protection. Coming to our for this, we are trying to find centennial blocks using sufficiency. For building sufficiency, we are using Correct algorithm. Correct algorithm is pretty good and works in linear time. After building sufficiency, we are trying to find LCS and MEMs. LCS refers to longest common served string and MEM refers to maximum unique matches. And further, we are performing deep depth first search on sufficiency and we are identifying LCS and after that we are finding MCLs. And after this step, we are filtering our short matches. Like some matches may be very short, which are not biologically relevant. So we are removing all those kind of stuff. For short matches, we are going to match matches which are similar to each other to form larger and conserved patches. After that, we are going to after we are going to find Levinston distance, which gives the amount and the minimum number of changes required to convert one genome to another. And we are also using Jacket X to find similarity of uh, genomes that we are going to use. And lastly, for visualization part, we are using dot plots and scenery maps. Coming to the current status, I'm already in the sectors of extremely important all and it's doing pretty well. But it's for very consuming. And we detected items and engines. After that, I already triggered about short matches as I mentioned in the earlier presentation. And after that, I also use matching method to match and matches which are similar to each other to form larger concern strings. Some also planning its first based in the uniqueness and biological benefits. Which I will update later because I have this that I will add this because it's a bit complicated and coming to visualization part. I'm using forwards in Intellimax and then coming to and I'm also communicating distance with the check distance. This idea will not take much more time. I may need 2 3 weeks. That's all. Now coming to the coding part. Here, what I would like to tell you is I am not using real life genomes because I need some more time to test it out. Basically, I want to complete this project and directly test the final part on the real life genomes. So I am using some random genomes here, as you can see. Now I am also combining those genomes with some end markers. Why I am doing this? Because it's easy to compute suffix tree for a generalized string. So I'm using those markers and combining those strings to compute suffix tree. Now I'm calling a class called. I am creating an object for a class called suffix tree. Basically, it returns a suffix tree and it is stored in this variable called tree. After that, I am passing those the root of the tree combined text, which is this one, and threshold 5. This threshold 5 has some importance that I would like to tell later. Then it returns what we can say a list of strings to this part, to this matches. So again, we are sending those matches, means we are sending those list of strings to match blocks. Here an interesting thing happens that I would like to explain later. So it returns a list, and this list. Now we are printing. Uh, Now we are printing the elements in this list. What does this elements refer? These elements are nothing but our potential synthetic blocks. Now, what happens in suffix tree? In suffix tree, class suffix tree. Basically, like, now we are initializing all the necessary things, and this is a method called build suffix tree. Here, this runs on the basic principle of Cohen's algorithm. Basically, here we are doing nothing but we are activating we are initializing some activators so that I'm calling this as activators because here see active node, active edge, and active length. These helps to track where are we in the way which they help us to track in which part of the tree are we working in, and remaining all works to keep track of like say remaining number of suffixes to be added which are in this current phase, or else like which helps to keep track of and global and all the stuff. And the most important part of the code comes here, extend suffix trees. Here we share suffix trees, we have a text, we have a root. So what is the next? What's function? The next part is nothing but how are we extending the tree? The tree is extended, we are extending the tree in three parts. Let's say this part has one first part, and this complete code and second part. In this second part, we have two subparts. Let's say this is 2A and this is 2B. Now I'm going to tell you how we are actioning the tree. While we are working with suffix trees, there is a case where the edge is not matching. In that case, what we will do is basically, we are creating a new list for the part of the text what we are working with. And what happens if there is a match? Suppose for a we are getting a match, then we will give this current node as text to the parent node. So it becomes a children. So we are increasing the depth of the tree. So here we are increasing the active length by one, which this active length indicates the depth of the tree. And what if there is no match? Suppose when we are comparing what if there is no match, it mismatches. What happens? Nothing. Basically, we are creating or splitting the node, current node. That's how we are extending the tree. This is how opponent's algorithm works. And now coming to the part, collectible strings. Here I told you there is something important like threshold equal to 5. I said threshold. I told you there is an important function for this threshold. What it is? In the presentation, if you see, I told you LCS and MBS. Basically, we are finding longest common substrates. Longest common substrates are nothing but they are identified by using this threshold functions. Basically, this whole function recursively traverses across the tree and finds substrates which has a length greater than or equal to 5 and passes to this merge blocks. And again we are passing that to this merge blocks. What this blocks do is it what this merge blocks do is it removes all redundant or overlapping trees. Suppose, for example, in the list it contains GCT, GC, T. Here, GC and T are substrings of GCT. So, what is the need of using this GC and T? That's why we have to remove all redundant or overlapping substance because it's already there in the mainstream. That's what this merge blocks is doing. After we perform this merge blocks function, then we are getting our potential syntonic blocks. What I'm going to do is, no, I am also planning to give priority score. Priority score based on length, uniqueness, and biological relevance. This helps our model to be more impactful. That's what I'm trying to improve in this code. And I'm also trying to calculate Levenstein distance, which is nothing but edit distance. Basically, you are given with two substrings, two strings of genomes, and they are asking the minimum number of changes that you would make to this genomes such that one genome will convert into another. For that, we use Levenstein distance and dot plots to visualize all the conserved regions in our genomes. And I'm also planning to use Jacquard index to check the similarity between the two genomes. That we'll see later. Coming to the solution. With basically in this model, we are trying to find certain laws using some expression. Why we are finding certain laws is that it helps us to understand how evolution is when comparing to the organisms with common ancestors. And after we are finding assets and evidence, after I applied filtering and imaging techniques to improve biological relevance. After that, I'm going to calibrate instances to basic using extend steps to tell how many changes are required to change one setting to another setting. And the time using visualize it's like plots and secondary maps to visualize conserved regions. One references are using the material provided in our courses. And I am also following the multiple literature work like Open, Kurtz, Chen, and I'm also following Meglin's work. And I'm also referring this field algorithms for strings, trees, and book basis for computational